Dum-de-dum. Good morning, everybody. Oh, beat your rose. Let's get this out of the way. All right, while we wait for people to roll in, there's plenty of time for questions about anything and everything. So I am your question receiving servant. Morning, Tanya. Invited lecture, yes, that is next Tuesday, the 9th. So I'm still waiting on the Zoom link. I'm just double checking, make sure I haven't got it already. Don't think I do. Oh, I do have it. Awesome. Okay, great. So that has been sorted out. So I'll send and I'll double check to make sure I have the current copy of the feedback form, and I'll post all that up. This week's kind of crazy, probably over the weekend, to be honest. And then I'll post another reminder on Monday evening. Actually, you know what? I'm just going to make myself a reminder to post a reminder. Got so much going on, pardon me, right now, that if I don't use my calendar, then I'm kind of screwed. Oh, that's right. I know. I remember now. The one on Tuesday I have sorted out. It's the one the week after that. On the 18th, I don't have the information for yet. So yeah, I'll post that. Actually, make myself another reminder. Sorry. Honestly, if I don't kind of put it down, then I'll just forget. Other stuff will kind of grab my attention. Put that. All right, I'll do that today. So in my other class, and my guess would be that all the people that are currently present in this Zoom session will be present in the invited lecture, right? Because they're here. But for the two that I've done for my other class, several people didn't realize that it was in a different link, different Zoom link. So don't use the one for this class next Tuesday. Use the one that I send to you this afternoon and then on Monday to get into that invited lecture. So you have to be present. Well, obviously you have to be present, fill out a feedback form, because otherwise you're not going to know what the lecture was like, right? But you have to be both present and submit your feedback form. Now I'll be setting up. Have I done it already? I can't remember whether I'm coming or going, to be honest, at the moment. Let me just double check, see if I actually have done that or not. Come on. Oh yeah, shit, I've done it already. I'm sorry. I'm a gibbering mess right now. It's already done, right? So if you, I should show you what my calendar looks like. It's bloody crazy. Yeah. Come on. Yeah, this is what my calendar looks like this week. Get that out of the way. So it's just crazy. Anyway, so at least for next Tuesdays, right? You won't see the one on the 18th here because again, I don't have that information yet. But next Tuesday, this is where you'll get out of it. This is where you will find the feedback form. You can see it just there. And this is also where you'll upload your feedback form after the lecture. So these are really useful bits of information for us, right? We do read through all of them, and typically, students' kind of opinions seem like kind of trend fairly closely to what the faculty think, right? Because we've been doing this for a while, but every now and then we get extra insight that we wouldn't have got otherwise, right? Particularly about whether you think that would be a professor that you'd want to do research with, or if it's a professor you'd want to take a class with, right? So that information is very useful and will be used in the process of choosing who we offer the job to. So it is worthwhile. It's also actually really cool because you'll get to see some, well, what I hope, based on the two that we've seen so far, some pretty cutting edge research. The one yesterday was just like, kind of, holy crap. I've learned more about this than I have in like 3 years of teaching it. Her work was all about microRNAs, right? So regulatory RNAs. Anyway, so you need to be both present in the class, in the invited lecture, using that link that I must have already sent you, but I'll send it again anyway. And you must also upload this form by midnight the following day, so next Wednesday. You could do both of those, and it doesn't matter what you write in here. I mean, I'm not grading you based, it's not like an essay. Just the fact that you have uploaded it, that is sufficient. And those attendance and uploading it will get you your 10 points for that invited lecture. Let's bring this back. Any questions about that? And actually asking questions to the candidate is also a good thing. Makes them feel better because they feel like they've had a receptive audience, and it's a good way for you to interact with scientists, right? So people that most likely you will be joining in the field in some form or other in the future. Okay, so what have we got to do today? So we need to finish going through that chi-squared cross, right? Working through that let me, where are we at? Tuesday, Thursday. That must have been the 28th. No. It was this one hiding here. All right, I'm just going to zip that up a little bit so we don't see Tanya's arm magic. Right, so we need to finish working through this. We need to finish off the probability stuff that was actually at the beginning of the probability and statistics PowerPoint. Just basically addition versus multiplication, independent versus linked kind of events, or exclusive events, as is the better word. And then it's going to be a busy day. We've got to make most of our way through the mitosis PowerPoint, and so there's a ton of stuff that needs to be gone through there. Okay, so how many did the chi-square cross? Come on, fess up. I see one hand raising. Oh, actually, I only can see one student. Did anybody else do it? Come on, fingers crossed some of you did. Crickets. All right, so I'll work on the assumption that you did, and I'll go through it anyway, right, just to make sure. So we have had a bunch of data. Where are we at? Here are the data here. Crab sticks, I can't actually see stuff. There we go. Right, so we had, our ratios were 960, or not ratios, our data. 150, 220, 350. Right, and that was dominant, crabs, sorry, dominant, dominant, recessive, recessive, dominant, recessive, recessive, in that order. Okay, and we also worked out the expected, right? So this was from a dihybrid cross. This is F2 generation. So our expected values would be 9/16 of the total, 3/16 of the total, 3/16 and 1/16 of the total, because we always have in our F2 generation a 9:3 to 3:1 ratio, right? Obviously, if we're doing a different kind of cross, we'd have different expectations, and therefore the expected values would be different as well, right? So our expected, what is that? That's 945, 315, 315, because again, you get 2 3/16, and scroll down, 105. All right, so whip out your calculators. Let's work out what those values are. Yeah, check. All right, so thank you. So what's the chi-square value for the dominant? So remember, observed minus expected squared over the expected, right? So that's, okay, awesome, thank you, 0.238. Now you need to do the same for the other three. So it would be 150 minus 315 squared over 150. It would be 220 minus 315 squared over 315. And 350 minus 105 squared over 105. Right. All right, we need those values. And in the meantime, oh, thank you, Leviette. Is that the total? Is that for the second category? I hope I pronounced your name right. Is that Leviette? Oh, for the second one, thanks. It's kind of a little bit like communicating with like astronauts on the moon. There's always like this little lag time because it takes people to type stuff out. All right, so got two values. What's the third one? Okay, right. So let's add those together. That's our sum, right? So the chi-squared value is the sum of all those calculations. So you have to add those together, and I'll bring up our chi-squared chart. Okay, so what's the total value? I'll stick that over there for now. Awesome. All right, so that's a pretty huge value. Okay, so let's figure out how many degrees of freedom we have, right? Degree of freedom, how do you calculate that? What's the like little mini formula? Right, it's n minus 1, where n is the, or yeah, is the categories, right? The number of those sums. Exactly. So the degrees of freedom will be 3 because we have 4 different categories. Okay, so now degrees of freedom 3. We're looking at this column. The number needs to be larger than this number here, the 7.815, for that result, that difference to be significant, not due to random chance, or not very likely. And so our chi-squared value of 687 is like way off the chart over here, right? And actually, if you use funky style stuff, if you use R to calculate that, you actually get, this is the actual p-value down here, right? So that's point 0.0000000000000002, I think. Maybe I missed out a 0 there or added an extra one in, right? So that's a incredibly significant result because that chi-squared value is huge. And so really what that chi-squared value is telling you is that the gap between your expectations, right, based on your understanding of what you were doing and your results, your observed data, was huge, right? And so clearly your expectations were wrong. Okay, Rose, let me go back. Oh, that actual value, Tanya, you'd have to tell Rose how to do that. She's going to give like a little workshop later, right?
It's given by like a program that's called R. So you put in the information and it automatically tells you what the exact value is. So you save a lot of work with R. It's kind of lazy work.
So that's why Tanya can work out chi-squareds in like 0.03 of a second. Whereas the rest of us, yeah, the rest of us are used our index fingers. And you do it manually as well. That's also good. I mean, it's kind of like using Excel to calculate different descriptive statistics. You know, it's a useful tool. I'm sorry, got the sniffs today. So, but Rose, for our purposes, we don't really need to say any more than our p-value. Well, our chi-squared value is larger than this value here by some significant margin. But really, statistically, all we need to say is that our p-value is less than 0.001. Right, so we would write that. So essentially, our kind of output would be our chi-squared is, let's scroll up a little bit, 687. Degrees of freedom is 3. And our p-value is less than 0.001. That is the formal output of a chi-squared test. And so if obviously this is not even if it was carried out as a dihybrid cross, some of the assumptions that we're making about that dihybrid cross must have been wrong, right? And so I actually kind of made the numbers what they were. Where are we at? Let's kind of go back a little bit. The chat bars look super twitchy with my mouse. All right, so these are the observed data. These are the expected data. So essentially what you're seeing here is a underrepresentation of the non-parental phenotype combinations, or an excess of parental. Essentially, it's the other side of the coin, right? Same thing. And so if you saw this in the output of an F2 of a generation of a dihybrid cross, that automatically would tell you that these two genes are linked. They are not assorting independently because you have fewer of these, right? You can see it's a 9, should be 933.
We're
Shit.
not, sorry, we're not seeing, I guess your screen, we're still on the probability table, the chi-square
Oh,
table.
you can't see the chat. Oh, I thought you could actually see everything that I do. Cool. I can do stuff you can't see. So if you look in the chat box, kind of imagine what I'm gesturing at. If you scroll up to where I wrote out the observed and the expected and the 4 different phenotype combinations, just scroll up a little bit. It's just above when Rose said bless you when I sneezed. Useful marker. I'm sorry, I didn't realize you couldn't see the chat box the way I see it. Can you see what I'm pointing to now? Okay, so essentially what you have there, I mean, if I was to do it properly, then there should have been fewer of the double homozygous recessives, or the double recessive. But what you're seeing there is an underrepresentation of the non-parental combinations, right? The dominant recessive and the recessive dominant. And that's a classic sign of linkage. So those 2 genes are located somewhat close together on the same chromosome, and they are not assorting independently. Don't stress too much about that now, because that's a whole nother topic that we'll get to later on in the course. But it's kind of, again, to put a worm in your ear about this when we get to it later, if that makes sense.
Would the same thing be if they're like represented as in the homogeneous recessive? Would it be the same situation?
No, because if you have linkage, you'll only see fewer of the non-parental combinations. I can't. If you see deviations from the 9 to 3:1 ratio, it's either linkage, if these are underrepresented, or it's epistasis, where one phenotype combination masks that. Well, one phenotype masks that of another. So things like Labrador coat color is a classic one. Actually, cat fur color too is another one. We can talk about those later. And those depending on the type of, if it's dominant, get out of it, dominant or recessive epistasis, it depends, it changes the ratio. So one is 12 to 3:1, and the other one is, oh shit, what's the other one? Yeah, 12 to 3:1. I can't remember what the other one is. I'd have to go back and double check. I think it's 9 to 4:3 or something like that. But yeah, so if you get one of those disappear, then that's a good sign that epistasis is occurring. And again, that's something we'll cover in the non-Mendelian genetics. I don't want to spend too much time on it now because we've got to cover other stuff and we'll be covering it later. But it's really just to kind of get you thinking about how we use this to test our assumptions. Cool. Okay, so any questions about chi-squared tests? Just going to mute my audio for a second. I need to blow my nose. Figure you don't need to hear that. Okay, so let's finish up the probability stuff so we can crack on with the mitosis subject topic. Okay, so essentially we kind of skipped ahead a bit because I wanted to spend some time on this and make sure I got it done with you before the weekend. But there's a few other little bits and pieces in here which are worth covering too. And did we cover, again, my apologies, I can't remember sometimes exactly what we covered between different classes. Did we cover the multiplication rule and the addition rule? We did? Oh, sweet. Oh, that's cool. We don't have to do that again. Anybody got any questions about it? So quick recap being that if different events are independent and you want to see what the probability of both of those events occurring is, you times the probabilities together, right? So a boy and then a boy would be a half times a half is a quarter, right? To have two boys. If one of those events, you can only have one or the other, and that particularly applies to order of events, then those events are exclusive, right? So if you roll a die, you can't roll a 1 and a 6. You can only roll a 1 or a 6. And so in that case, you essentially have, let's say, two chances of getting the number you want. And so you add those probabilities together. So if it's a roll of a 6-sided die, it would be 1 plus 1/6 plus 1/6 to get one of two different numbers. And that's pretty much it. And that also kind of really applies to this, looking at the F2 generation of dihybrid cross, right? These different genotypes, right, are exclusive. The phenotypes are independent, right, because they belong on different genes, right? So you can only have homozygous dominant or for one gene, or heterozygous or homozygous recessive. You can't have both for one given gene, right? So those genotypes are exclusive. The phenotypes, however, are independent, right? You can have both green and round, or yellow and round, or yellow and wrinkled, or green and wrinkled, right? Those are exclusive, so you times those probabilities together. Okay. All right, so if anyone has any questions, pop up. Oops, let's not do that. Put that back where it was supposed to be. If not, I will move on to mitosis. Okay, cool. So, my apologies. I think it's allergy season right now, so I'm a little bit bunged up. Okay, so two big reasons for talking about mitosis, and we'll always talk about mitosis first. Well, overall reason is because it's how you end up being you, right? So you started as, pardon me, one single cell, fertilized zygote inside your mother. And then by the time you made your merry way down the fallopian tube to the uterus, you probably divided, hang on, 1, 2, 3, 4, I think like 6 times. I think it's about 64 cells, give or take. Looks like a little blackberry. It's kind of cute. Like yeah, it looks exactly like a blackberry. It's called a mulberry stage. I don't know, perhaps the person who discovered it liked mulberries more than blackberries. And get this, this is a super cool fact. Did you know that you actually hatched? This is a fun bit of information. So we always think, we don't, we're not like chickens, right? So we don't have like hard eggshells, but you do actually, oh, cool. That's good to know, Chance. We're more up on the physio side of things. But you actually were an egg at some point. And so as you kind of bounce your way down the fallopian tube to the uterus, you most definitely don't want to stick, right? And so the eggshell around you, or was around you, kind of lasts until um, about the 64 cell stage, give or take, by which time you're in the uterus. And then you actually hatch out of your eggshell. How cool is that? So there you go, a little bit of fun physiology fact. Anyway, so obviously there are hundreds and hundreds more rounds of cell division, and as time goes on, those cells become more differentiated, right? They become specified into different types of cells, be it, get out of here, bone cells. I mean, early on it would be, skin progenitors versus gut progenitors versus neural progenitors, and then so on and so on and so forth. And so after however many of those, I don't know how many rounds of cell division occur, you are a multicellular functional individual, right? And then you obviously continue that process through childhood to end up more or less how you are right now. And then you start decaying slowly into old age, which is kind of sad. So we don't talk about that very much. So the mitosis is a basis for you being a functional multicellular organism, right? And so that's the big picture reason why we talk about mitosis. But the detailed reasons are twofold. One, we get to talk about the cell cycle and the various checkpoints that are involved in that, which are extraordinarily important. And also, we get to practice this stuff for when we go through meiosis, which is considerably more complicated than mitosis, right? For a whole bunch of reasons. And it's actually where most of the really exciting stuff in cell division happens, right? It's kind of mitosis works with what you get, right, to make you who you are. Meiosis is what gives you essentially who you are, right, in terms of your combination of alleles that make you, different from your siblings, your parents, your grandparents, and so on. So let's move this over a little bit. So you can't see this. You can't see me like waving the chat box around on your screen. That's so weird. Okay, kind of funny. All right. So yes. So essentially, when we talk about the cell cycle, what we're talking about is going from a cell which is not currently dividing. That may be because it's just finished dividing. It may be because it was what we call quiescent, right? Which is a funky word, which basically means kind of like sleeping. So quiescent. Well, really, the technical term is not dividing, but kind of sleeping sounds better. Right, and so that's this G0 over here, right? So essentially a G0 cell might be a cell that's just recently divided and hasn't yet got the signal to divide again. It might be a cell which is already, on the path to differentiation, right? So it won't divide again, right? Those called post-mitotic, like neurons, for example, are post-mitotic cells, right? They never divide again once they've been differentiated into neurons. Or it could be like a stock of, let's say, stem cells, right, which you have scattered all through your body and your skin, gut. Skin and gut are the two main ones. Hair, like hair follicles, for example, that are just waiting for the signal to enter the cell cycle again, right? And so that cell cycle proceeds from that point when that cell gets the thumbs up from whatever, some other cells around it telling it what to do, all the way through what we call interphase, which is the preparation phase. So this cell has already committed to cell division, right? It's not just resting there, even though it looks more or less the same, is actively preparing for the final part of the cell cycle, which is the M phase, right? Which is the bit that we actually call mitosis, right? So one heads up, you have to be very, very careful with language, right? So mitosis in this case can apply to the whole process from entering G1 to cytokinesis. That's often how it's used, right? So we're talking about mitosis right now. It can also be used very specifically to talk about the division of DNA in the cell cycle, right? Which is just this little bit here, right? In the M phase. So you have to be careful exactly what you're talking about when you say mitosis. And I'll be also, when, not if, when I ask you questions about this, I will be very specific about what I'm asking you about, right? So that I'm clear about which I'm talking about. I tend to refer to, when I use the word mitosis, I tend to refer to this particular part, but sometimes I do just kind of slip into the vernacular, right? And refer to the whole thing as mitosis, right? It's just bad habit, I guess. So anyway, this interphase part is really serves two purposes. One is it prepares the cell for nuclear and cytoplasmic division, right? Which is mitosis and cytokinesis. And it also serves as a series of checks to make sure that this cell really should be dividing, right? And these checkpoints are, there's the G1 to S. Is that hidden? Yeah, there it is. It's hidden behind the chat box. This G1 to S checkpoint, the G2 to M checkpoint, right? So basically these are checkpoints that control whether or not the cell enters the next phase of whatever, right? So the G1 to S is towards the end of, well, I'm never entirely clear exactly where it is. In the G1 phase, I need to do a lot more reading on that to understand that better. But it controls the transition of the cell to the S phase, right? And we'll get what these phases are in a little bit. Similarly, the G2 to M is actually at the end of the G2 phase, the second growth phase. And this controls whether or not the cell enters mitosis itself, right? The division of DNA. And then the final one is here, and this is another super important checkpoint, which is a spindle assembly checkpoint, or SAC. So these checkpoints are, when they function correctly, and they don't always function correctly, but when they do, these are ironclad guardians of the cell cycle, right? These are like the, I don't know, the stone knights outside of Hogwarts, right? That are there to protect the gate, which actually is one of my favorite scenes from Harry Potter, you know, the whole film series, is when those start jumping down. Anyway, that's a bit of a tangent. I don't tend to quote like classical literature when I try and pretend to be intelligent. I tend to quote kids' films. But anyway, by the by, these three checkpoints are ironclad, right? If they don't work properly, bad things happen, right? And typically, if either of these two fail for whatever reason, due to mutation or other stuff. So, quiet please, love. I'm teaching. Actually, does anyone know what might happen? Right, these are checkpoints that stop cells dividing, essentially, until they're supposed to.
Isn't that where it's mutation and then like cancer cells?
That is exactly where one of the points at which cancer can progress. Right, so a classic, I mean, there are lots of ways in which cancer can occur. You need that signal, to tell the cell to divide for a start, right? And those signals tend to be developmental signals, which should be off, right? So excess signaling is one problem. The other one of the other ones, at least, suppression of cell death is the third one. But the second one is these checkpoint proteins don't function properly, or these checkpoints as a whole, either through mutation or they may be modified by other proteins. They're actually extraordinarily complex processes, to be honest. And if they don't work properly, those cells that are currently kind of in the holding phase can enter cell division. And depending on whether these other checkpoints work, they can actually continue all the way through. And so that uncontrolled cell division essentially leads to tumorigenesis, right? And obviously there's a lot of other different things that need to kind of go wrong essentially for cancer to progress, which is a whole nother topic that we don't really have time for, unfortunately. But these checkpoint proteins, or these checkpoints, are like they have to fall or fail, right, for cancer to progress. And so, go on, Tanya.
So, sorry, I have a question. So, from what I understand, the cells, when they're like not functioning well, they're gonna like kill themselves, right? The apostasis or something like that.
Apoptosis.
So, or yeah, that. And so new cells are gonna form because they're too old and they're not functioning. So why would we age? Wouldn't like new cells produce like skin cells? Because I'm in my 30s, so
oh well, you're a spring chicken then in that case.
I think so. I'm going downhill now. So why wouldn't like I thought like cells would like new cells and don't get like wrinkles?
So that's an interesting question. Part of the issue is senescence, right? Which is where cells kind of cease functioning properly, but they don't enter apoptosis. So apoptosis is, I mean, apoptosis by definition is a programmed cell death, right? And so quite often apoptosis is a planned part of development. So for example, your neurons, you have about half the number of neurons that you were born with, because all the ones that you don't need were killed. It seems a little harsh, but it's called, neuronal trimming, which occurs during early childhood. Sometimes apoptosis occurs because, so this is like intrinsic apoptosis. It occurs because something's gone wrong inside the cell. So, for example, poly-Q disease, muscular dystrophy, things like that, Huntington's disease, right, are the result of protein aggregates building up that the cell can't clear or get rid of. And so at a certain point, those become toxic and they die. Overexcitation of neurons also can kill them as well. Neurons are pretty fragile cells, to be honest. So that's kind of like internal to the cell, but also there's cells which can tell other cells to die. And that's where a lot of the tumor suppressor proteins are involved. Like you've probably heard of P53 is a classic one, for example, or c-Myc is another one, right? And so those actively tell cells which they find dividing when they shouldn't, that they should die. So those are all fairly clear, defined things, whereas senescence is just like cells getting kind of old and crappy, right? They just don't work as well and they kind of just get shitty, basically. And so it's not really enough to trigger apoptosis. And also your ability to replace those cells kind of reduces over time because there's a limit to how many times cells can divide before they hit a limit called the Hayflick limit. And so essentially, it's kind of a whole bunch of different things. It's not actually a topic that I understand super well, to be honest. but part of it is kind of cellular senescence. Part of it is your decreasing ability to replace cells as they die. Um, and also some cells can't be replaced, right? So trying to think, neurons are the classic ones, kidney cells, for example. Trying to think, eyeball cells, so your eyesight gets worse as you age. Part of the reason for that is, you can't replace those cells, at least not very easily. And so it kind of, all those things kind of tie together, and like the accumulation of damage over time through like free radicals and the like. Um, and epigenetic marks, so on and so forth, so that things just work less well. So there you go. Sorry.
All right, thanks. It was just something like it didn't make sense for me, but now it does.
Right.
They're cells, but they're not too bad to just kill themselves, so they're there hanging.
Right. Yeah, and it's a pretty like cellular suicide is pretty serious, right? You don't really, as a cell, you don't want to do that unless you have to, or unless you're told to. And so uh, those cells don't reach that threshold, basically. Um, yeah, I mean, Ali, that's also a good point. So you can accumulate mutations over time, and also from DNA replication, and also external insults like carcinogens, essentially. But actually, in many ways, it's not the DNA that gets damaged; it's the marks that get added to the DNA. These are called epigenetic marks. So methyl groups and the like, right, which we'll talk about later on. And those are, they basically control, or they change how genes are expressed, right? So you're less likely, say, to produce a protein which might keep the cell functioning properly because its expression kind of decreases over time. That's also an interesting point, and again, we'll get to that later. Why identical twins become less identical as they get older is because they accumulate those epigenetic marks, because obviously their lives are different in some ways, and you can actually tell identical twins apart more easily after middle age than you can when they're young.
Professor, but wasn't it like DNA, the telomeres and the chromosomes, like with stress, they shorten, and that's why you age as well, or something like that?
They
Like
don't
stress
shorten with
affects.
stress, they shorten with replication.
Oh,
And when we talk about DNA replication, we'll go into the mechanism of why that happens. And after a certain amount of cell cycle, it's about 60 times dividing, unless you're a stem cell and other stuff is going on, then you hit a natural limit. It's called the Hayflick limit. And once a cell hits that, it can no longer divide, and it basically begins senescing after it's been around for long enough. I mean, if you think about how long your red blood cells live for, admittedly, your red blood cells go through an awful lot of like wear and tear, but they live for about 3 to 5 days. The neurons that you have, right, and those are like super long. You can't actually see how far apart my arms are because my webcam isn't wide enough for you, right? Those exist since before you were born until you die. The same cells don't change, right? So I always think that's kind of amazing. Anyway, I think we need to kind of, wander back onto the path. Yeah, biology is just amazing. It really is. It's a constant discovery. That's why I love teaching, because I have to learn about all this stuff so I can teach it to you lot. But anyway, so essentially the cell cycle is a series of processes and steps, right? Actually, what have we got down here? Do we have any notes? Nah. So essentially the first checkpoint is a couple of different things, right? Broadly speaking, right? I'm not going to go into the mechanisms by which these checkpoints work because I kind of understand them. Actually, I'll work on some of them for some of my research. Um, but we don't really have time, and I don't think I could do a good enough job to really warrant going into it. But there's a whole world out there if you're interested to kind of dive into. But this checkpoint, pardon me, checks for essentially nutritional levels, right? Is there sufficient energy to make it all the way through? And that's important because once a cell begins dividing, it has to finish. If that cell does not finish dividing, it will die. Right. So once it enters that process, it's committed to it. So you really want to make sure you got, the provisions and wherewithal to make it through to the end. Right. So, and actually, this is one reason why people that are severely nutrient limited, so growing up during famines or in poverty, why they tend to be smaller. This is kind of like part of the cellular process behind it, right? So they undergo less growth because many times their mitosis is checked because there is insufficient resources for that to proceed. So that's actually part of a bigger kind of phenotype, one of the ways in which it acts at a cellular level. So the other thing it's checking for is damaged DNA. And so if the DNA is damaged, and that's actually what BRCA1 and 2, BRCA, which are essentially, I've probably heard of Angelina Jolie. She had a double mastectomy because she carried mutations in both of those BRCA genes, and that kind of gives you like a 70 to 80% chance of breast cancer. And also, there's a strong familial history of breast cancer as well. And so the BRCA, I'm not sure if they act in both of these two or one or the other. I don't know them well enough. Those check for damaged DNA. And they try and repair it, right? But if they're unsuccessful, then this checkpoint is blocked. And that's because you don't want to go into the DNA synthesis stage here and copy DNA that is damaged. Because either DNA replication will not proceed successfully, or you will copy that damage into the daughter cells, which is also not what you want, right? Does that make sense? Yeah. So then you go through, and so this is essentially accumulating all the various bits and pieces needed for DNA replication. So all the proteins and the resources and so on and so forth. We then copy the DNA. So we have sister chromatids. So those are identical copies of a chromosome. That's not, and that's kind of versus homologous, which are essentially not quite identical copies. And you have two of those. Right, so that's a little bit of language. It can trip people up. So essentially, in all of your non-dividing cells, you have two copies of each chromosome, right? We're diploid. We have 23 chromosomes and we have two of each, so we have 46 total, right? Those copies are not identical, right? If you kind of look at here, essentially they have potentially different alleles for each gene. Don't always, but if you look at the chromosome as a whole, they will be, ever so slightly different. Sister chromatids, on the other hand, right, are identical copies, right? So the process of DNA replication turns this one chromosome into two linked identical chromosomes, right? These are the sister chromatids. These will then be split later on during DNA division in mitosis in the M phase, right? Important bit of language. Anyway, once that happens, then the cell starts preparing for mitosis and cytokinesis. So it's just kind of building up a bunch of stuff, basically. And we have our second checkpoint here, which is another DNA damage checkpoint. And that's essentially to make sure that all of the chromosomes were replicated completely and properly without any major damage during the S phase. Once that's given the go-ahead, then we enter into mitosis, which we'll talk about in a little bit. And our final checkpoint is to make sure all of our chromosomes are lined up on the metaphase plate. So this is kind of what this looks like. Right, so once all those chromosomes have been pulled to this, and it's not an actual line or a plate, right? It does actually become a line later. This is the plane of cell division, right? The actual cytokinesis. But at this point, it's simply those chromosomes get pulled to the center of the cell simply because they have equal tension pulling on them from both sides. So if you have, you know, a person in the middle with holding on to two ropes and the people at the end of those two ropes pull equally, right? Then that person in the middle should be exactly between the two people at the end, right? Essentially. Right, and so what the spindle assembly checkpoint does is it checks to make sure each chromosome is in the center of the cell, right? We don't have a chromosome up here. We don't have a chromosome down here. We don't have some which are busy like zooming around all over the place because they haven't figured out what they're doing yet. We have them all lined up along the middle here. Once that occurs for all of those chromosomes, and it has to be all of them, right? Because you can't allow anaphase to go ahead if one of your chromosome pairs is in the wrong half of the cell, you know, one half. And so once all of those chromosomes in the middle, essentially the spindle assembly checkpoint says we're good to go. And the glue holding together these sister chromatids dissolves and those sister chromatids are pulled apart. Come back up here. Told you I kind of bounce around a little bit when I do this kind of in the Zoom session. And then as soon as that occurs, or kind of sort of alongside it a little bit, then the cytoplasm gets divided. We now have two identical daughter cells, right? These are not different, even though they're like in different parts of the diagram. These are exactly the same. They have exactly the same DNA. They have exactly the same, more or less, cytoplasmic contents. These are identical daughter cells. That's one big difference between mitosis and meiosis. Mitosis, the two daughters that are produced are the same. In meiosis, they are not. Does anybody have any questions about the cell cycle? It's a bit of a wild ride, I'm afraid, but we're going to need to make our way through it. Okay, so another thing that we need to talk about a little bit is kind of chromosome structure, right? So we have, do we have anything about Shigoshin here? Oh, I want to talk about Shigoshin. Sorry, I'm going to just type this in because it's cool. I looked this up this morning. I kind of knew what it was, but I found a cool fact about it. So Shigoshin is something that prevents the of cohesin, if I can spell it right. No, don't autocorrect. And so cohesin is the glue that holds the sister chromatids together until they shouldn't be together, right? Which is in anaphase. And Shigoshin is, this is the fun part. It's the Japanese for guardian spirit. How cool is that? I wish I got to name things like that. I don't know what I'd call them. I don't think I have enough imagination, to be honest. But so this is part of that spindle assembly checkpoint. So this is essentially the bit that stops anaphase from occurring until all of those chromosomes are lined up on the metaphase plate. And so another thing that's important to talk about is, so we have essentially cohesin, right, which is kind of stuff that holds these two sister chromatids together. We have our centromere, and a centromere isn't an actual thing, right? It's just a region of DNA where the kinetochore will assemble, right? So it's not, if you look, I mean, there's a little bit of difference in structure, but there's nothing that goes, that's a centromere, right? It's really just a region of the chromosome which will result in the assembly of the kinetochore. And this kinetochore is really a bunch of proteins which bind to that region of DNA, and they serve as the anchor points for the spindle fibers, for the spindle microtubules. Those are the ropes that will pull on either side of that chromosome, that pair of sister chromatids, until it's kind of jiggled around and it's in the center of the cell, right? So these the kinetochores are essentially like kind of like the, if you think about like a suspension bridge, right? They're the sockets in which the cables go into to hold everything up, right? That's essentially what they do in cell division. Okay. So, any questions so far? No? There's quite a lot of information to get through, so I appreciate this might be a bit of a kind of a deluge kind of right now. Okay, so once we've gone through, oh, another fun fact as well. The P arm of the chromosome is always the short one, the short arm, because the centromere isn't always like exactly in the middle. And the P stands for petite, which is French for small. I don't know what Q stands for. I think they just used Q because it sounded nice. That's my guess. Anyway, so mitosis, right? If we're actually talking about the mitosis part of cell division, is the process of dividing the DNA equally between the two daughter cells, right? And it's obviously an extremely important process to get right, because otherwise you end up with one cell having too many chromosome copies and the other one having too few, right? Which is a very, very bad thing to happen, right? It does actually happen in human reproduction. You're probably familiar with Down syndrome, right? Which is trisomy 21. That occurs through meiosis, in which we'll talk about that next week. But it's a similar kind of principle. So I always remember this as PMAT, because my name's Matt. I don't know. I'm not going to talk about what the P is useful for, but anyway. So essentially that stands for prophase, metaphase, anaphase, telophase, right? P, and there's also another one in there, which is kind of like an in-between stage called prometaphase, right? So a mnemonic you can kind of, well, not a mnemonic, but kind of an acronym you can think of maybe is like P is for preparing, right? Is the process by which, you know, everything gets ready. Metaphase is when they meet in the middle. Anaphase is when they pull apart. And telophase is when you end up with twins, right? So that's another way of kind of thinking about it, right? Whatever works, right? I really don't mind. It's just what helps me remember stuff. So essentially another thing that's worth remembering too is that these are prophase and telophase are bookends, right? So they are the opposite of each other. Prophase is when things fall apart and chromosomes condense and spindle fibers form, things like that. Telophase is when the nuclear membrane reforms, when the chromosomes decondense, when the spindle fibers fall apart. So whatever happens in prophase, the reverse must happen in telophase, right? That's a pretty easy way of remembering those two stages, right? So in a sense, you don't have to remember what happens in prophase, and you know what happens in telophase. It's just the opposite. Metaphase is the lining up of the chromosomes. Anaphase is pulling apart of the sister chromatids. That's really all there is to it. So the mitosis or the M phase isn't actually super complicated. There's a whole bunch of stuff that goes into it. But in and of itself, the different stages are not too tricky. So essentially in the last part of interphase, our chromosomes have been replicated, right? So that we have every chromosome now consists of 2 sister chromatids. Our centrosomes, which are our anchors, essentially. They do other things as well, but basically they act as the anchors which are going to be pulling on the chromosomes. Those have been replicated. What else has happened? Yeah, all the other doohickeys, proteins, stuff like that that are needed have also been made, right? And so once the cell gets the go-ahead to enter mitosis, the M phase part, then a whole bunch of things start happening, right? And so this is kind of prophase and prometaphase. These are, I think they're distinguished by kind of distinct events that occur, but they're really a continual process, right? And so essentially in prophase, the beginning of that process starts, right? So these centrosomes start pushing themselves apart so that they will end up on either side of the cell exactly. The chromosomes start condensing, right? So it's like a kind of bowl of spaghetti, right, at the end of interphase. Even though these chromosomes have all been replicated, it's like you've kind of got, 50 strands of spaghetti and another 50 strands of spaghetti, and you just mix them together in a bowl, right? So that's obviously extraordinarily hard to organize and work with when you're trying to pull those things apart. So one thing that is really important is that those chromosomes start condensing, right? They start kind of wrapping themselves around each other. They get organized so that they basically go from this crazy, crazy, crazy long thing, and they kind of shrink down into, the classic, I don't know what you do, the X-shaped chromosome, right? This process then continues, right? It's essentially, it's not like a hard break. And that nuclear envelope, the nuclear membrane falls apart, has to fall apart, because otherwise, how are you going to attach the spindle fibers to the chromosomes, right? There'll be a membrane in the way, right? So the nuclear membrane has to disappear. The chromosomes condense even further down to those kind of stereotypical X shapes, right? Which you can see here, right? So these are, well, these aren't, I don't think these are X shapes. I'm not entirely sure, but these are super condensed, right? They're not how they look normally. And these spindle fibers, right? So you see the process that began here, these centrosomes moving to the opposite sides of the cells. That process is now finished. So these centrosomes, or they're also called microtubule organizing centers. So MTOCs, if you want to sound fancy. These are now anchored to the inside. They're like kind of like stuck, right, to the inside of the plasma membrane on either side. And they grow these spindle fibers out. Kind of always think of them like harpoons, right? It's like kind of whale hunting in the dark. You know, they fire these out, and some of them may find, just by random chance, a kinetochore, right? So you see one that's attached here. These ones, you know, whoever was manning the spindle fiber gun at that point wasn't very good and fired them off in the wrong direction, right? So these don't attach to anything. They're still important, but they don't attach. And so this process just keeps happening, like boom, 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 boom. Takes a while until you have a spindle fiber attached to the kinetochore on both sides of that chromosome. So essentially you'll have a spindle fiber attached to one sister chromatid, and you'll have a spindle fiber attached to the other sister chromatid. Can you see my cursor, out of curiosity? You can? Oh, that's good. I probably should have asked that a little bit earlier in the course, right? Anyway, these ones here only have one. So if you pull, and these spindle fibers want to contract, right? If you pull on those, these 3 chromosomes will end up at the top of the cell. These 2 chromosomes will end up at the bottom of the cell. That is bad. You do not want that to happen, right? Because that'll mean that whatever this chromosome is, this daughter cell that will form from down here will get 2 copies. This one up here will get none, right? So this whole process goes on until every single one of these chromosomes, these sister chromatid pairs, has a spindle fiber attached to both sides. And then, you know, they're pulling on both sides, right? And that kind of chromosome gets jostled around until it ends up in the middle of the cell, right? And that has to happen for all of them. Now, this whole process takes a while, right? If you look at cells that are dividing, most of them will be in prophase, prometaphase, or telophase, right? Once this occurs, once metaphase, they all line up, essentially that spindle checkpoint or spindle assembly checkpoint goes, yes, 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 blink, and it activates a protein called separase that cuts the bonds holding those sister chromatids together. It dissolves cohesin. And as soon as that cohesin, that glue is gone, you have tension on both sides that's kind of pulling, right, around at those sister chromatids. And the glue goes, watch it, Tanya. This is the exciting part. Ping! They get pulled apart. Just basic mechanics, right? It's physics. It's actually biophysics, which is kind of cool, right? It's not something that's I don't know how you describe it, but basically it's those ropes, those spindle fibers are slowly broken down and they're kind of reeled in, and they pull the chromosomes to the opposite sides of the cell. Then all that happens is the opposite of prophase and prometaphase. Yes, I'll take you out for a walk soon. My doggo's giving me a mournful look. If you can see her. There she is. She's like, I want to go for a walk and sniff stuff. And that anaphase happens very quickly, right? It's just mechanical. And once it does, then you just need to basically fix up the mess, right? And put things back to how they needed to be. And so those spindle fibers will disappear. The nuclear membrane reforms. The chromosomes decondense. And that metaphase plate, right, that line, right? So the chromosomes were lined up here, pardon me, equally between those two centrosomes, those asters, right? And so they actually leave behind proteins, which then basically tell the cell, this is where you are going to divide. And so those proteins, it's called a contractile ring. And so it's basically like you get a balloon and you squeeze it in the center, right, to make, two balloons if you're a clown, let's say, right? And that contractile ring gets smaller and smaller and smaller and smaller and smaller until pinches off, and now you have two daughter cells. And that's cytokinesis, which is the technical word for cytoplasmic division. And that's pretty much it, right? So this is what that cleavage furrow looks like in animal cells, right? Quick question. Oh, all the Pearson stuff is for lab. So that's some question to ask Mr. Penny about. I don't assign any extra work other than the lab assignments for my lab section. The work for this lecture, which is due tomorrow. Shit, what is today? Thursday. Saturday night, sorry, is the first homework, and that's in Blackboard. Yeah, the Pearson stuff is for the lab. I don't know anything about that. And plants have it slightly different because they have a cell wall, right, which is a kind of like a physical structure, right? It's not a amorphous, flexible structure like the plasma membrane in animal cells, although plants have plasma membranes too. But what happens here is they actually kind of build the wall essentially between the two cells, and that then causes division. So rather than the pinching off kind of process that animal cells go through, plant cells, because they're rigid, they have to be, they build up the wall instead along that metaphase plate. And that's pretty much it. The only other funky thing that's worth talking about, which we'll come to in meiosis ever so quickly, is that in animal cell division, say this is a zygote that's divided for the first time into two daughter cells, every subsequent division is at a right angle to the previous division. So these two daughter cells, right, we've got the plane of division kind of north and south here. These two daughter cells, their next cell division will be essentially the line, rather than being like that to provide two, now you're going to have a line like that to provide two above each other. So the plane of division rotates 90 degrees every time. Whew. That's a bit of a wild ride. Thank you all for hanging with. So that's it for today. We have the invited lecture on Tuesday, and we have meiosis, which is way more complicated than mitosis, on Thursday, and then the following week is our first exam. So hopefully I'll be getting plenty of questions from you all, my email or next class, or not our next actual class. Also, if any of you have questions that you can't easily formulate into an email or something that you want to work through, then let me know and I can schedule like little mini Zoom sessions with you individually to talk about the course content so that you can get fully prepared for the exam in 2 weeks' time. So I do have, even though my schedule is crazy, I do have time in my schedule for that to happen. And that's essentially kind of part of this whole process. Okay, so I've gone over a little bit. I'm going to call it a day so you can get off to your next thing. I'm going to take the dog for a walk, and I shall see you next week. Have a lovely weekend.